This video illustrates a protocol beginning with the injection of a fluorescent protein-expressing tumour cell line into the mouse mammary fat pad. A specialised chamber for in vivo imaging is constructed and then surgically implanted into the animal, at which point imaging data can be acquired.
Hi, I'm Dr. Bojana Gligorijevic from the laboratory of Dr. John Condeelis and Dr. Jeffrey Segall of Biophotonics Center at Albert Einstein College of Medicine. Today we will show you a procedure for intravital photoswitching and imaging of Dendra2 labeled cancer cells through the Mammary Imaging Window. We use this procedure in our laboratory to study influence of the tumor microenvironment on the motility of tumor cells. So let's do it!
Wearing latex gloves, begin the fabrication of the Mammary Imaging Window, or MIW, by heating up a 5 to 10 cm tissue culture dish and creating a curved surface by pushing a rounded hard object, such as a 1-inch Dremel bit, against the dish. Cut out the centre of the dish using a heated razor blade. Now use a small, cone-shaped Dremel bit to make a 6 to 7 mm diameter hole in the centre of the dome-shaped plastic base. Make the edges of the plastic base completely smooth by sanding it with the Dremel and further filing it. File the top of the plastic base to make a flat surface.